a boot camp type session, so that we're not just talking about this, but maybe showing some practical tips and hopefully having enough time to answer some questions to make sure that when we leave this session, we have at least a few people, hopefully everyone on the call, who has an idea of either a project they maybe want to develop from scratch, or some projects that they hear about that are ongoing that they maybe want to get involved in to sort of see what it's like as a collaborator. But either of those, I think, would be great outcomes from this meeting. Next slide. Thanks, Susie. So I just want to point everybody's attention to the INRG website. Probably most of you have already been there. Specifically today, we're all presenting on behalf of a large international group, the INRG Strategy Development Committee, which really has multiple goals, but the main goal is to get new people, young and not so young, but new people involved in projects with the INRG, not just involved, but also to help us think of new ideas as well, and to help mentor people in this process. And then the website, specifically under the Get Involved tab, has a lot of the past presentations that we've given to various YI and other groups that will have some more details on things that we won't pass over a little quicker today, because we know we have these other slide decks, because we really want to get to the more hands-on participation part of today. So next slide. So in case you haven't done this, it would be great if you can click on your participant name and enter your name. If you want to, you could enter maybe your country or institution, but at least if you can have your at least first name or first and last name, that would be great. If you have a question during the main part of this or during the Q&A, feel free to either put your name in the chat and say you have a question, or put the question or clarification there. And hopefully the rest of our team who's not speaking can help bring it to our attention. And reminder, you've probably already heard this, that the meeting is being recorded for those who maybe could not make it today. And to please, of course, mute yourself. I think everyone's got good Zoom hygiene at this time after all this time. Next slide.
Meredith, just one more note. We're going to send a link to the questionnaire to gather more information about people.
Great, thank you for reminding me, Susie. At the towards the Q&A session, I think what we'll do is send you a link to just get a little more information about all of you, and like I think 2 questions about what you thought of the sessions today, so that we can maybe work to always add more helpful, pragmatic information in the future and figure out what the next step will be. Next slide. So we're your hosts. As I mentioned, we're a part of the Strategy Development Committee, and really focused specifically our little group today on how to get you involved. So just briefly, you see all our pictures, and we don't have time to kind of go through long intros, but I'm Meredith Irwin. I'm from the COG, based in Canada. Lucas is from Spain, in the SciUp-N. Mark is a YI out of Chicago, who is going to do a lot of the practical part of how you can get involved, as he has. Lieve is from the Netherlands, also participates in the SciUp-N as well as other groups, and has a strong interest in mentorship. Wendy is our lead statistician for the INRG, and has such an amazing, great understanding of the opportunities and some of the challenges using the INRG database, and I know can provide great insights to all of you today and potentially during project investigations. And Susie, who you've already heard from, is really super responsible in a very amazing way for helping organize today, and almost everything about INRG could never happen without Susie. So she's almost the leader of this whole session today. I just want to thank her for all of her help. And she really wants to get more people involved. And she's a great resource, even if you just don't know who to contact about something. Is that okay to say, Susie? Great, thank you. Next slide. So this is just the agenda of what we'll be doing. I'm not going to go through it in detail, but you'll see we really want to have the bulk of the time, half of the time, for demonstrations, for Mark to talk about how he and others would recommend you frame your research on some practical tips on how to use the database. And then towards the end, just the practical part of starting an application and some projects we've started to think of that could use either collaborators and or leaders, and questions, of course. Next slide. So just briefly, we'll hit on some of the existing projects. And again, these are really listed in great detail on the INRG website, as well as in some of the previous slide decks from this SDC committee. So I don't want to go through it in detail, other than talking in broad strokes. I'd say there's two main areas that in the INRG group we think about. One are consensus statements papers, which of course are enabled by the database, but examples of those are the risk classifier, which we're in the process of revising, as well as some consensus papers on biomarkers, making sure we all speak the same language for risk factors, those types of things. And then the majority of projects, more than 35 approved to date, are really classic data mining or clinical or translational projects where someone has a question that either cannot be answered in smaller cooperative groups or smaller multi-institutional studies, or would be much more powerfully addressed by the INRG large international database. And so I would ask you to look at the website, because when you look there, you may actually get an idea of, oh, nobody's looked at this, or this was a great idea. Here's another way I can look at a rare group or some other type of question that you have, especially maybe started in your own clinical practice. And so these are just a list of a few of the ongoing projects. And the first one is just to remind me, the neuroblastoma drivers one, that there is the potential for some questions to link to some other databases like the Target Genomic Database and others. The second example is one of the maybe more classic data mining, looking at the role of gender as a prognostic indicator, and that is being led by YI. And then more recently, several of us are involved in the new risk classifier work that we're doing with the newest parts of the INRG database. Next slide. And just briefly, I will introduce the data dictionary, and then you'll hear much more about this from Mark in his presentation. But essentially, the data dictionary is the type of data that is collected and inputted by our amazing statisticians in the various cooperative groups. And it includes multiple clinical and bio risk factors, as well as pretty extensive outcome analyses. And the details for each variable is in this presentation. And again, we'll make these slides available so you can even link on through this slide deck to that. But I think it's really important to look at this to determine the feasibility of the projects. And the next slide summarizes some of the issues to think about when you think of the data that lives in the database. And the left side is the data that's available, and the right side is data that everybody always wants, including myself, for projects, but is not currently available in the current database, but might be data that could be available in smaller studies with various cooperative groups. So on the left, there's the typical risk factors we all know about, the clinical and biological risk factors, and you can see them listed there. We do know treatment categories, and the details of that are in that previous slide deck. So we'll know things like if you were on a study that randomized between two different transplant regimens, we'll know which trial arm you were assigned. We may not know if you actually received that therapy, but we'll know you were on the trial and that was what you should have received. And as I mentioned, there are links to some genomic data and very minimal scans that have been uploaded, as well as we hope at some point soon a digital path. And then people are always talking about new things to add, and I'll talk about that briefly before I hand over. But on the right are some really important ones we always get asked about that are not available. So we currently don't have a way to validate that someone received the treatment that they were assigned or that it was delivered. Surgical outcomes and details are not currently collected. Response data, so PR, CR, PD, those sorts of things at various time points are not currently available. And for ongoing trials, we may have some data, but we will not have outcome data until those studies are published. And that can sometimes be a limitation. For example, if you want modern cohorts, there will be some of that, but the numbers won't be as rich as they will be from older cohorts. And then metastatic site information is available pretty routinely for the SIOP-N patients, but only for a subset of the COG ones. Next slide. So these are just things to let you know are coming. And since most of you are younger than many of us presenting today, there will be great projects to think about. And it's actually never too early to think about a project and potentially get approval for a project that you might want to do when this data becomes available. So Lucas, with a task force, including Julie Park and others, and Wendy, are working on figuring out how to incorporate relapse data from at least subsets of patients treated in some of the cooperative groups that are part of the INRG. So that will be a really great data set in the future to ask powerful questions. Preclinical data, so if people have xenografts and they've done data related to patients on trials, that's not currently available, potentially one day, although there's not a real active group working on that. Response data, again, in small pieces, will be at some point available, but is not in the next couple of years likely to be available for large numbers of patients. And I already mentioned the linkage to some rich genomics sets. The next two slides, or I think my last two slides, just to give you a taste of some of the genomics and biology factors that we're moving forward to sort of incorporate. So on the left here, you can see different factors, and there's pages and pages of these different data that are collected in the data dictionary. But I'm highlighting here on the left, the last few rows. And maybe the next slide just shows you where we're going with those. So currently we're able to collect 3 loci, 11q, 1p, and 17q, just whether they're lost or not. And moving forward, what we want to do is collect more rich data on more loci, as well as whether they have a segmental chromosome aberration pattern or numerical chromosome aberration pattern. And there's a group on the genomics biology committee working on how we will recode that particular data point. And I do think, optimistically, that data will become available for subsets of patients in the next couple of years, maybe sooner. We've worked through a lot of the early logistics of that. And then for ALK, we also currently are able to collect whether there's a mutation or amplification, or if we don't know. But moving forward, we'd like to get more detailed information uploaded about the particular variant, the % of cells potentially having that if that's available, as well as where that sample was taken and the time point diagnosis or ultimately other points. So those are early, but they're high priorities for our biology committee to incorporate working with the informatics team at INRG. So things to start thinking about, questions you could ask across large populations. Next slide. So at this point, unless there's any burning questions that we wouldn't get to later, I want to pass it on to Mark to really give a practical look at how one can get involved. And I have to say, Mark and previously Sam Volchenbaum, who's really one of the main leaders of this database, have been very helpful in helping me to come up with questions that are feasible and how to proceed or make my question more feasible moving forward.
Great. Well, and if no questions, I don't see any hands raised or flags. Thank you, Meredith. I also want to point out we have 2 people on this call who I think are also probably in many ways much more knowledgeable about this than me and the inner workings and the history. Wendy London, she set up this database, she framed it, she developed all of these field codes. So Wendy, please, if I say anything that needs clarification or correction, please feel free to interrupt. Brian Ferner, also on this call, is a main point person at the Pediatric Cancer Data Commons. He works very closely with Sam Volchenbaum. Brian is really the guru of getting data in, quality controlling it, and getting data back out. So he's sort of the day-to-day operational person that makes data happen in the INRG. So again, Brian, if there's something that I'm saying that's maybe not quite right, please feel free to comment and weigh in as well. So I wanted to start just by sharing the data dictionary. I know we've talked a bit about it already, but this is the way I was sort of trained with databases. So I got involved in database work working with Steve Du Bois when I was a resident. He actually started me on the SEER database, which is another NCI-funded database. And he was the one who said, this is a great way to get involved as a young investigator and try to answer some interesting questions. And that subsequently led, when I came to University of Chicago, to working with Sue and sort of asking sort of similar types of questions, but in a sort of a bigger way with the INRG and in a very neuroblastoma focused way. And one of the ways that I was trained to sort of think about databases is, well, what are the data that are there? And Meredith did a nice job of highlighting some of the big picture things, but I wanted to get into a little bit of the nitty gritty, because you can only ask questions of the data if the data exist. So taking a step back, how do the data get there? Well, the data come from cooperative groups. And how do the cooperative groups get the data? They get the data from people enrolling on studies. In the case of COG, any patient who's enrolled on the biology study or a clinical trial And in the case of SciPen and many of the other consortiums, is people who are enrolled on clinical trials. And that information comes from sites, hundreds of sites across the world. And each site is reporting that data to the cooperative group on a case report form, associated with either the biology trial or the clinical trial. And that data then has to get processed by statisticians within the cooperative groups in some way to send to the INRG. So when we think about what is in the INRG, remember that there's many layers of data transfer that have gone in before it ever gets there. So if you say, oh, well, wouldn't it be really great if I had this one little bit of information or this other sort of piece of data? Remember where that data came from. It came from all these hundreds of sites submitting CRF. So if that was never in a CRF, or if that's hard for the statisticians to get out, you're never going to have that in the INRG. So I really, especially for people who are trying to think about their first question, try to think about in the context of what exists. And as Meredith said, what will exist in the future. So what the data that are here are the data that are here. And don't try to think about, unless you know what other data exists in some other database you can link it to, what else you can add, because if you ask the exec committee, can I get this data, even though it's not in the dictionary, the answer is going to be no, by and large. The couple of things I want to put on the data dictionary. So this is, if it's here, it exists. If it's not here, it doesn't exist, right? But one of the things that's really important about the INRG data dictionary is this thing called the USI, or the Universal Specimen Index. This is a crucial patient identifier. So what happens, right? A cooperative group will send their patient's data. They'll get a COG number, a SciPen number. That will actually get converted by an honest broker to a USI number. And then everything linked to that patient goes to the USI number. And that is a way to link the INRG to other external databases. If you're interested in linking patients with INRG and Target or Gabriel Miller data or any other sort of external genomics data set or other types of data set that are out there, this is how a patient will be tracked with the USI number. So this is a really clever way that was devised a while back to track patients in a de-identified way. I mean, the nice thing too is when you're telling your IRB, what am I going to do? Well, I've got this de-identified USI number. It's completely unlinked from the clinical trial number or anything else, and I have absolutely no way of identifying it. That really speeds up an IRB process to be able to say this is 100% de-identified data. Of course, you got standard things, as Meredith mentioned, in terms of age. You get year of diagnosis. There's this initial treatment field here, and some of it, when you're thinking about databases, is what is sort of intrinsic and what is a derived field code. A derived field code means you sort of use other piece of information to come up with another piece of data. And this initial treatment is actually a derived field code. So these are the things that you learn when you work with the database, and you don't have to remember this, but I want to point that out because there is some of that in here that when you talk to a more senior mentor, they will likely know. This is just saying, well, because they were treated in this year and they had these risk factors, we think this is likely the therapy they were to have received. So take this particular field with a grain of salt. I think always this is inferred. You'll get information, on initial trial. If you're trying to figure out who was assigned to what therapy, that can always be helpful. There's things like stage, and you can have, whatever staging system you want, Evans staging, INSS, INRG. But again, remember that depending on when the patient was diagnosed, they may have 1, 2, or, unlikely to be 3 of these different stages. The older patients in the database may have Evans staging and INSS. The brand new patients might have INRG and INSS. It'll be a mix. And that's something that, as you sort of think through your statistics and you think through your code, that you get to take into account. MICAN amplification, ploidy, these are sort of classic biologic features. You can tell that somebody who works in a statistical program made these field codes because you have 11q with an underscore under it. Most statistical packages don't like your variable name to start with a number, so hence the underscore. So thank you, Wendy. And we have a little bit of information on ALK, but again, remember, the older patients in the database aren't likely to have ALK information because that wasn't collected on those older patients, and there's no way to go back and sort of find that again. Other lab values like ferritin and LDH, older studies may have collected this. A lot of the new COG studies, and I don't know about the SciPen studies as much, you may or may not have that field on any given patient. There's a lot of information on site of disease in terms of where the primary tumor was, and a little bit about where the metastatic was. But again, this is a very much a yes/no kind of question. Was it reported or not? And then, of course, anytime we're doing an analysis in the INRG, most of what we're going to think about is outcome data. Did this group of patients do better or worse than that group of patients? And so it's obviously important that we have things like event time, event censoring flag. Did the patient have an event, yes or no? And then their follow-up time. And the nice thing about the data dictionary is it will tell you exactly, how an event is defined according to the INRG. And that is the nice job of the data dictionary. There is, very little ambiguity on, how are they defining an event? How are they defining death? How are they defining INPC? I will admit that I often have to go back and sort of refresh my memory on what like diagnostic category versus histology versus grade means. But it's all nicely, clearly laid out there, so you can map it back to whatever biologic feature you're interested in analyzing. One of the things, and here you'll have things like overall survival time, cause of death, race, ethnicity. I know COG reports this. I don't know if SciPen has as much of the race and ethnicity data. So, depending on what any sort of group might have collected, you may or may not be able to do a whole sort of international study. Oftentimes we limit to COG studies or to SciPen patients because one group collected a certain piece of data while the other one didn't. And that can be a very valid inclusion or exclusion criteria for your study. If your primary variable of interest is X and X wasn't collected by the cooperative group, you have to exclude that, patients from that cooperative group. And like I said, please feel free to interrupt me, ask questions. I can't see questions, so somebody please stop me if there are questions. Also in the database, as Meredith mentioned, there's things like treatment arm. So for patients on 032, A3973, so right now these are all COG trials, although I believe there are efforts to get SciPen trial information in here as well. Information on second malignancies and where was the second malignancy and what it was. And then, a little bit more on what treatment arm. And as Meredith said, this is treatment assigned. One of the limitations of any INRG paper, if you see written up right now, is we don't actually know what treatment the patient received. So these are the field codes that exist, and it's limited to 35, 40-ish different fields. And one of the things that I think is always interesting that you don't see here is something like risk. So risk is not actually part of the INRG. So you cannot go into the INRG and say, is a patient low risk, intermediate risk, or high risk? I mean, that's obviously a sort of a very clinically relevant question that just doesn't exist in the database. And I think there's a very rational reason for that, which is risk groupings change, right? So, if you use a modern INRG risk classification system versus an older COG risk classification versus an older SciPen classification, most patients will probably be the same, but there will certainly be patients that were intermediate risk, if they were classified 20 years ago, who will be high risk now and vice versa. And so part of, what you need to think about as somebody using the database is how, if risk is important to your analysis, how am I going to assign risk? Which classification system am I going to use and how am I going to code that in my statistical software? And that's another thing to think about is oftentimes in these database analyses, you can derive other interesting field codes and risk is one of the things you can derive. You can use things like MICN and stage and ploidy and the best of your ability assign risk. And because this is not a randomized clinical trial or even a clinical, a single arm clinical trial, you get to define risk however you want. And as long as it's justifiable to your peers and to the INRG exec committee, that's okay. And you can, you have the freedom to make those decisions. Again, as long as you can rationalize them scientifically. And the same thing with any other field code you might want to derive, whether that's, a split in age, whether you want to define it as age less than or greater than 18 months, these are all things that you can do with the data, in a statistical software program. Questions about that so far? I see Sam joined us as well. So the other thing I wanted to highlight too is what happens when you actually get data from the INRG. So you've come up with a question, you've taken a look at that data diction, you said, okay, these fields exist. I can sort of think about this grouping of patients in this way compared to other patients grouped that way with the various field codes that exist. I've created my primary variable of interest. This is sort of most INRG studies. I want to compare group A to group B and sort of see how they do. I've applied to the INRG, and I think Lucas will go over that in a second. I've gotten approval. Now, what do the data look like? The first thing that's actually really nice about INRG data is that it's quality controlled. So Wendy and Brian primarily are responsible for this. They will take all the patients in your requested cohort and run them through these quality checks. And this is sort of anytime you do a database project, you want sanity checks, right? So they look and they say, okay, did anyone have, because remember, this is all coming from case report forms. And as we all know, there can be data entry errors. And you don't want to include data entry errors as much as possible. So they have gone and looked and said, okay, is anyone labeled as having metastatic disease, but they also have stage 1, right? I mean, those two things are incompatible fundamentally, and they will go and flag those patients. And for the most part, the things that are really egregious for a database, like stage 4S disease, but a 5-year-old, those patients have either been resolved or removed entirely from the database. So the nice thing, and you can see sort of this whole list of QC. This is as of a couple of years ago that this QC was done on a particular project. You'll get a report of how many patients matched each particular issue. Whether they, none of them thankfully match these sort of red and yellow criteria, the things that are really bad for any particular patient. We had 344 patients who had some QC issue, but as you can see, most of them were in the sort of green range where they maybe had stage 4 or 4S disease, but you didn't know what their metastatic site was. It was labeled as unknown. So they could still very much have metastatic disease. You just don't exactly know where that metastatic disease was. And depending on the questions you're asking, that may or may not be important. If your analysis is dependent upon site of metastatic disease, you'll need to remove these patients. But if you're looking at something totally separate, like clinical trial enrollment, this may not matter. And this is where you get to work with your statistician to figure out, are these patients appropriate for this analysis or not? And they've given you the choice. Whereas the things that are really egregious, they've sort of thrown out those patients because those are probably not quality data. So again, this makes it really nice that these data are very much plug and play. When you get the database and you get the data, you can put it into a statistical software package and start running. And with that said, sort of what do the data look like? I mean, I think I just wanted to show briefly what that might look like, because I think that's often sort of demystifies the whole process in some way for people who've never really looked at a database. I've completely randomized this, so these USI numbers are garbage. You can't actually use them for anything. You will notice that probably not all patients were, less than 100 days when were diagnosed in 1991. So, this is an example of what the data might look like, but this is obviously not quality data or something that anyone should use. So this is what you will get. So you say, I want all these patients to meet some criteria, and I want them from year 1991 to present or whatever, and you will get exactly what it looks like in the data dictionary, right? So you'll get their ID numbers, you'll get their age and days, the year they were diagnosed, all these things, and it's zeros and ones for was it primary pelvic site or was it a 9 for unknown. Stage will be 1 through 5 typically, or unknown. 5 being stage 4 because there's stage 2A and 2B, and so on and so forth down the line. There are some messier field codes in here, so you'll get things like cog number. You also might get things like initial trial. Initial trial is a messy field code for people who are interested in statistical software and all. This is a database of several thousand people, but you'll have things like this, where it's, patient was on 9047, 8742. This person was on 3 or 4 different trials. Many of the, this was a person diagnosed a long time ago. So some of these trials might be a biology study. Some of these trials might be a therapeutic study. Some of these trials might be a long-term survivorship study. That's sort of on the researcher to look that up and figure that out, again, depending on what the question is you want to ask. If you don't care about what trial they're on, then it's less important. But if you're trying to do an analysis based on was this person treated on some trial, it's something that you just have to code out, whether it's you yourself or your friendly statistician. But again, I think for people who have looked at databases before and have used statistical software, I know that, if you've seen a PCDC or Pediatric Cancer Data Commons presentation, there's a lot of work being done right now into putting everything and harmonizing the data and changing the field code. So I should say that some of these things may change in the coming months as some of these field codes get integrated across diseases. But hopefully the end product will still be an Excel spreadsheet. As sort of old school as the Excel spreadsheet, I guess technically this is a comma separated file. This is really easy to put in statistical software. You can take this and paste it into Stata, into SAS, R, you name it, and start doing analysis basically right away. And then, if you know any coding yourself, that's always helpful. But of course, that's why statistical support is really important. Because your statistician, when they look, most statisticians, if they look at a file like this, will say, I can use this. This will be plugged. I can easily fix the null variables into something that won't cause issues with this being a string variable, and and do very, very minimal cleaning. Again, I always appreciate the efforts that Wendy had put into this, because this does actually make it very much plug and play kind of data once you have come up with the idea, gotten the approval, and actually receive the data. So that's pretty much what I wanted to say right now. I don't know if anyone had any questions or comments, but I hope this gives people at least a little bit of an idea of what an INRG project actually looks like and what the database actually looks like. Because it really is, in many ways, the output or the input for your analysis, or the output from a data commons, is this flat file that's readily easy, fairly straightforward to use.
Thanks, Mark. I think that was a great sort of practical introduction in what you've used. And maybe we'll move on to Luke, because I think I could think of a few areas we might want to elaborate on in the Q&A if people have questions, especially how to determine if there's enough patients to answer a particular question before they move forward. So maybe Lucas.
Can I just make one mention?
Please, thank you.
Yeah, it was really, really excellent summary, Mark. Thank you so much. This is just in doing analyses, keep in mind that the overlap across patients for non-missing data can get very small. So if you take 20,000 patients in the INRG and you say, give me all the patients who have where all their data are known, that's only 500 patients. So in any given analysis, you have to be careful about the overlap of the number of patients who have known data, and work with your statistician to find a way to deal with missing data, because it is going to be a big problem in the analyses. But the key variables, the most important ones, are non-missing in most patients. It's just that overlap across patients, the non-missing data number with non-missing data becomes very small.
That's really important. And another thing, like the treatment, that we get asked for every paper, I think, from the INRG. Thanks.
Great. So thank you very much. It's really great to have you all here. So I'll try to give a brief, some tips on how to get started, because many of you have reached us through the email that we launched, and what happens next? How do we move forward? Here's the initial thinking process that we would need to follow. How do we start an INRG project? Well, one would have an idea. Before this committee was created, one had an idea, already went to the application to contact the INRG executive and have the application. And we thought that it would be really important to have these two intermediate points to exploit them a lot better. So one of them is that you can get in touch with us in this email address. Meredith and I get the emails, and we will try to help providing some mentorship. We will find colleagues within the INRG community that will provide some help and mentorship, and maybe give some steer at the beginning until we make it to an application. Then there's the cohort discovery tool, that it's a tool available in the INRG website, but that it's also going to be available soon in the PCDC website, that allows you at least to find out how many patients of a given subgroup that you want. It allows us to do some small exploration to see how many patients with infants with MECAN amplification are there in the database, and what kind of data, or how many patients have linked genomic data. So that's helpful to know, and it will continue to evolve to make it more practical. But even without a specific idea, I think we're very happy to be approached because with interest in participating in neuroblastoma, we'll mention later a number of projects that are ongoing or that are starting. So we might help even shaping the idea. You don't need a definitive final hypothesis to get in touch with us. So next slide. Once we give form to the idea, we do need to fill the application form for the INRG review process, and it's in the website. It's a simple form. We need a group of authors, the young investigator that will be taking the lead on the project, but then ensure sufficient expertise on the topic that we're going to address. We do need a statistician, and I'll touch on this in the next slide. And then the project proposal is structured in several, like for any project that is starting in the aims and hypothesis, which patient cohort are we going to look at it, and what's going to change, the background, the justification for doing the analysis, and the data that is requested, and how. So if we go to next slide. In terms of statistician resources, all the INRG database projects require a statistician being a co-investigator, a collaborator in the proposal. And one can either collaborate with one of the INRG statistician committee members, or find a local statistician at one's university institution that has. However, this has to be approved by a group of statisticians from the INRG. At the beginning, that's something that we're trying to help also from this strategy development committee and from these initiatives. We might find help with some resources for new investigators, particularly for investigators starting their first INRG project, so that they don't need to find time in their own institution, or a statistician involved will help identifying these resources. It is really important to have a statistician in these studies, as Mark was highlighting, for all of the steps that are needed, because they will know very well the database. They will help with the design of the study. They will tell us what is feasible and what isn't, and then of course run the specific statistical analysis. I don't know if Wendy, do you want to make any comments? Or
Thank you for your kind words, and I feel very strongly that a statistician can help making the project successful and defensible when published.
Absolutely, yeah. A statistician is needed from the very beginning till to address all the reviewers' comments and finally getting it published. So we go to the next slide. So with the proposal, as mentioned, we're trying to assign a mentor to each of the new investigators and ideas, with a mentor that is familiar with INRG and familiar with the specific question that is being developed, and we will help with these initial steps, and that's why we have this the email address that Meredith and I review, and then the proposal goes to the INRG executive, or by a group of them that review the proposal. There may be some questions, and then in the end it's approved, and then the data is ready to be analyzed. If the data is going to be analyzed by a new statistician in a new institution, there has to be a data transfer agreement, but then the data is sent and the work can start. So next. And then, as we were saying, we're very happy if each of you comes to us with an idea. I really want to find out what's the outcome of these patients, group of patients, or to link this data with this other data, and that's absolutely great, and will help you get to the application and deliver the project. But sometimes when one is starting and doesn't know well the INRG, what can be done in INRG, how to collaborate, the people. you also have the opportunity of joining existing projects that we are trying to develop. So we have classified them in 3 categories. Some of these projects are well advanced, some of them are just starting, some of them are pending data to be incorporated. As Meredith mentioned, we keep adding and adding, and so more patients and then more fields in different areas. So if we go to the next one, the first group is reanalysis. As you know, the INRG started nearly 15 years ago. So the initial tranche of the initial group of studies were done with the first 9,000 patients. And many of these questions, now that the INRG has got 20,000 or more than 20,000 patients, many of them would be worth repeating the analysis, because now we have got a lot more contemporaneous patients that have been treated with contemporaneous treatment. So many of them may be really worth repeating. These are some of those that we thought would be important. So one is a paper that Wendy London and Victoria Castell did, analyzing factors predictive of survival after relapse. So that was done in 2011. There are tons more of new patients that could be analyzed and see if any of these factors have changed. And there's some people working on projects in this area. Then one of the key achievements of INRG is the new risk classification system that was done led by Sue Cohn in 2009, and that's again in progress by a group of several people, mainly Meredith, Matthias Fischer, and Wendy London, because that massive analysis can be repeated in a new risk classifier. Another paper that is a very good example of a young investigator, Daniel, that analyzed a subgroup of patients. So he found out the prognostic significance of the pattern and burden of metastatic disease in patients with metastatic neuroblastoma. So again, that was done some years ago. It is probably worth repeating the analysis and see if with contemporaneous treatment, with patients being treated with immunotherapy, this has changed. And again, some subgroup analysis. INRG is very helpful for some of the analysis in very small, in very rare groups of patients that cannot be done with the usual tools. So, for example, there's been an analysis of some stage 3 that were called stage 3 patients, also work with localized neuroblastomas or infant neuroblastomas with MECAN amplification. These are some papers that were done years ago and could be reanalyzed. Next. Another area will be the new analysis on data that is going to be added into INRG. So there's going to be new genomic data, and we heard about ALK, about segmental chromosomal aberrations, and other will follow, so that can lead to new projects. There will be new relapse data. We also mentioned with more details on the relapse patients and data from the relapse trials. Again, another new area. And also we're hoping to have response data finally imported into the INRG, the CR, PR, or MIBG scores. So these could lead to new projects. We have never worked on the impact of treatment modalities, as you saw. There are already some fields on the basic treatment elements that the patient had, but actually we really know what treatments they were assigned. We know which patients were randomized to 1 transplant, which patients were randomized to 2 transplants. We know which patients were randomized to immunotherapy, and we've never analyzed the impact of these treatment modalities. And then Wendy London has been working on a specific on the London risk classifier for patients at a clinical risk classifier for patients at low and middle income countries, so that you don't need expensive genomic analysis to classify the patients, but you just use clinical variables. And again, that's work in progress. So we go to the next one. I think that we're nearly there. That's finally, there are some projects that are not dependent on data. These are initiatives from INRG that don't really need individual patient data. So INRG is working on some white papers, consensus documents, and for example, Meredith is working with others in the group in a white paper on basics for neuroblastoma biology research. Also, Wendy and I are starting to work some systematic reviews of prognostic biomarkers that will inform future neuroblastoma research, but these are non-data projects where people could also be involved and help. I think that's the last slide. Yeah, I'll thank you very much, everyone, particularly Susie, as Meredith mentioned, is leading all our group to help young investigators, and we'll be happy to hear everyone's questions and comments and continue this dialogue throughout. Thank you very much.
Thanks, Lucas. And maybe while Liv gets set to sort of start a Q&A, if people want to put any questions in the chat, and also Susie has put in the chat a very brief, mostly multiple choice, 5 or 6 questions, just to help us know how we can help all of you. Because our goal really is to have at least a few people, and hopefully all of you in the next few years, get involved in a project. So I'll pass it on to Liv.
So currently, indeed, in the chat, there are only your comment, Meredith and Susie. But I guess there are many more questions with everybody. So one of the questions that came up is, are there any toxicity data in the INRG database? So maybe, Karin, you allow me to ask this question on behalf of you because it's your question. So are there toxicity data in the database?
No, there are not. And it's really, that would be closely linked with treatment administered. And we're really one step further away from that right now. We barely have a handle on what treatment was assigned to be administered, much less what the patient actually received. And there are currently no plans to collect adverse events that I'm aware of. It would be very difficult unless we knew how the patient was treated for those data to even be meaningful. So I don't see that on the horizon. It seems to me that analyses of toxicities are best conducted within the context of a particular clinical trial where you know where you can tell exactly how much treatment a patient received and what they, how many cycles they got even.
Wendy, can you remind me, second malignancies, how broadly are those collected? It's not really exactly the toxicity question Karen asked, but.
You're right. There is a data item for secondary malignancy. Mark, you can probably speak to this because you've done an analysis on that, but I am a tiny bit skeptical that is as complete as it should be. I don't know that we always get information on whether a patient actually developed a secondary malignancy.
Yeah, I would agree with that assessment, Wendy. I mean, remember, how are second malignancy data collected? A site has to actually report that to COG. I actually don't know that was ever collected in SAPN, although I think I heard maybe that's changing. And so it's likely that second malignancies are underreported in the database because, sites, within COG don't necessarily think to report that back to, the neuroblastoma B1 study that the patient was enrolled on years before. So it is there, and it is as complete as it is, but there is certainly a caveat in that.
Yeah, one can also think about long-term toxicity, so not the acute toxicity, but that's the long-term hearing problems or, well, short stature, etc. But I guess the clinical studies are starting to record this.
I think that
patients, so maybe these are future fields.
Yep.
Yeah, that's a good question. And right now in the COG, there is a study that recently closed, 377 patients with high-risk neuroblastoma who have survived at least 5 years are being studied. And that is a very broad database in terms of the number of data items about late effects and the granularity. And if someone has interest in that area, I think there will be opportunities for lots of manuscripts to come out of that database. And you can contact Tara Henderson and Lisa Diller, who are the PIs. I'm the statistician on the study, but I think that is the first real effort to begin to understand late effects. And until such time as those kinds of data items are routinely recorded on a particular clinical trial, I don't think they're going to be seen in the INRG, but these other databases specific to late effects do have very good information.
And I think the SciFN group is also trying to collect some of that data.
Okay, yeah, I'm sure they are. I'm sorry, I didn't mean to slight SciPen.
No, for sure. Just with some of our European friends joining.
Absolutely. No, absolutely.
We are indeed. So Teresa, do you have a question? Do you want to ask it yourself? Please go ahead.
Sure, thank you. Good thing I was not naked or anything. So I was just wondering, I guess, since you don't have any adverse events in your database, you don't have any quality of life data either. Is it something that you might consider in the upcoming years to collect as well? Thank you.
I think it's a great question. We don't. I think what has been entering into INRG is what is being recorded in the clinical trials. So even some years later. So as you know, frontline clinical trials are not collecting any PROMs or patient-reported outcomes or quality of life in neuroblastoma. In general, there's some initial validation studies happening. So I'm sure that once they start to be part of the routine of the clinical trials, we will be wishing to amend the INRG and incorporate them, because they would again be great to pool data and meta-analyze them, but they still need to be collected in the trials so that we can include them in the INRG.
Well, actually think about that, Susie, you may know more about this, but I believe the survivorship groups are starting to build databases within the Pediatric Cancer Data Commons. I think linked to perhaps like CCSS data.
Yeah.
So it's possible that through more of actually a survivorship group as opposed to the INRG, you might be able to find that kind of data at some point. I don't know when that will actually be accessible or launched, but within the greater data ecosystem, there are efforts to start centralizing some of that.
Yeah, the one other thing I might say just in follow-up to that, Mark, because you mentioned a few times the Pediatric Cancer Data Commons, which I put in the chat, is just if people do come up with questions that they might want to look at across more than one disease, which may happen, the Pediatric Data Commons may be a great place to think about that. And also, in case any of you are interested in diseases other than neuroblastoma, hard for me to imagine, but if that's possible, that's a great place to see if there's other databases where you might want to get involved in research too, just so that we're not all about neuroblastoma all the time.
Yes, I see a hand.
Yeah,
Emma?
I'm Ramya. I'm a pediatric oncologist from UK and part of the SciPen as well. So yeah, one of my questions was about the late effects, but I also had another question regarding teenage and young adults and adults. Has the INRG looked at this population of teenage and young adults or slightly older teenagers with neuroblastoma, and do we have any data on, say, for example, their clinical, biological, and outcome features? Or would that
Yes.
be something of interest?
Yes, that we could put that on the list of an analysis to be updated. If you look at Mose et al., I think it was a PBC paper, but it has been looked at, but that would be an older cohort, and I would definitely be happy to work with someone on updating that analysis with a more recent cohort.
Okay.
And even with more genomic data coming in, it might be
exactly.
more relevant.
So
Yeah.
it's Mosse, Yael Mosse is the first author, M-O-S-S-E.
I put the PubMed link in the chat for anybody who
There
wants.
you go.
And all
Very
of
That
modest.
is actually the nice thing about the INRG website, right? I mean, all these former analyses are right there. So I just actually went to the website and found the papers. So
you can
Yeah.
download the papers from the websites and abstracts and such. So it's a
Okay.
great
Which is
place to
very
start.
nice as well.
Thank
Yeah.
you. Yeah, that would be quite
Fiorella.
interesting.
Fiorella, you have a question.
Oh, yes. Hi, this is Fiorella. I'm one of the junior attendings at Memorial Sloan Catering. So I just have a question because I'm trying to get involved in projects with the COG and also the INRG. But I just, at the beginning, it's a little confusing which path to follow, or can you be on both? So I just wanted to hear a little bit more about that. I'm also super interested on looking at some potential collaboration for AI projects. I think they have, Mark has a project that he's working on, so I'm interested on potential collaborations with that and looking at data that way.
Fiorella, I think that the COG and the INRG would be thrilled to have participation from members of Sloan Kettering in both groups. You can definitely do both.
And I think the INRG mentorship is, it can be everything, but we're really focused especially on helping people develop projects so that it can still be around career development and other things. But I think our main focus is to help you take that idea or work on one of the ideas we've come up with as an opportunity and take it to completion. So for sure, both are great. And I think some projects are more feasible in one group versus the other. I don't know if Mark wants to comment on AI. I'm sure he does.
yeah, no, and so it's certainly, I think, an upcoming area with many potential pitfalls and hurdles to be overcome. But I think it's certainly possible, and actually something we are doing, to combine all sorts of data, right? That's the beautiful thing about many of sort of the newer genomic and AI techniques, is you can cross a lot of bridges. And the neuroblastoma field internationally is small, right? You get the sort of a lot of the same people again and again and again, leading a lot of different things. And once, as you, express interest and start doing projects and start, getting things done, people get to know who you are, whether that's through the INRG, whether that's through COG, SciAPN, whatever, which I think enables a lot of these sort of cross-platform collaborations and approaches. So absolutely, that's a viable strategy to be involved in all of the above.
So it might be, it's exactly, we're at the hour. So I know everyone likely has another Zoom or other thing to go to, but we just want to thank everybody for the time that they have devoted to this. And we really want to help you, whether that's one-on-one or with another session. So please take the time to the quick survey to give us feedback on what we should think about doing next, because we do want to follow this up. And we also want to be able to help you figure out a mentor. As an example, if there's an old project like the one Wendy talked about, where there was a YI who was maybe the primary author, it's possible they may be actually the best mentor to help with the updated analysis. So we can make those links. So please answer those questions. And we are incredibly optimistic that we might even be able to have some sort of a in-person, if we get to meet in the spring at ANR, with some of you and others who maybe haven't had the chance to join. So anything else to add, Lucas or Susie?
No,
Maybe
thank
I
you
can
very
add
much
that
for-
we really want to have
Yeah.
a hybrid meeting, so it's really virtual, preferably live at ANR. So we really hope that we will have also a live YI meeting there.
Hopefully.
And I just quickly put up all the other diseases that we're working on. You'll note that neuroblastoma and germ cell tumors are the most advanced in the amount of research, with soft tissue sarcoma coming behind, and the rest of these in development. And
Great.
my email address is pretty easy. It's suzi@uchicago.edu, and I'm just here to help.
And the INRG website has all these contacts, and we'll make the slides available on the website as well. And a follow-up email potentially about the next steps to all of you separately as well. But thank you, everybody. Again, this was great from my perspective, but we'll hear what you all think. And thanks so much to Mark, Lucas, Liev, and Wendy also, and Susie especially for all of their hard work for today. Thanks.
Bye-bye. Good to see you.
Bye. Bye.